Nationwide Children's is what we call a biospecimen core resource. There are two BCRs, we call them, in TCGA, and our role is really to get the best tumors that we can into the project. When these samples are first identified, and they're identified at a number of sites, we work with more than 100 different sites throughout the country, throughout the world. And once they get here, there are several steps of quality control that have to occur in order for them to go on through the process within TCGA. The first step is a pathology review, and so we have contracts with a lot of different pathology experts throughout the country, and we work with them to look at every sample that comes into the BCR and tell that it has a high degree of tumor in it. So we don't want to be looking at normal tissue, we want the specific tumor cells, and so that's a quality control check that we do up front. We also want to make sure that it seems to be intact, that it's not what we call necrotic, where there's a lot of dead cells. We really need high quality tissues for the next step of our process, which is to isolate nucleic acids, DNA and RNA, from each sample that comes in, and those then have to be quality controlled. At that point then we are responsible for sending that DNA and RNA out to the centers. There are sequencing centers and characterization centers at some very big name institutions in this country and in Canada. Our other role as the BCR is really to make sure that we have the clinical data that goes with those samples. So it doesn't do any good to find a novel, say, DNA mutation in 20% of your thyroid samples if we don't know how those patients did. And trying to correlate any new findings with that clinical data, and that's really going to give us our best idea of what we should do in the future.